So now we can go a little bit and read through this bioinformatics paper to see, now that you have been in this course for 4 days already, you will understand quite a lot. So let's go a little bit on this. So the title for this one, which is the data you're using, is Conserved Transcriptomic Profile Between Mouse and Human Colitis Allow Unsupervised Patient Stratification. I'm not going to details of the results of the paper, but I just want to focus more on the methods and that are used inside. Yes. So one of the things, it uses public data set, and now you know basically where it comes from. If you know GSE, you know it's from GEO, and it's publicly available. There are different data sets, and now you know that they are published in another paper. And one of the part of the results of the paper, so groups with no apparent subdivision, then we further statistically tested whether multi-cluster substructures were present in the dataset, since most clustering algorithms define transcript profiles, even on random noise. So you know that from the clustering analysis that random noise will be clustered if you ask to it. So however, bootstrap analysis using some another test to test if there's cluster exists presented a low clustering substructure trend, regardless of the gene ranking matrix used. The gene ranking matrix is either use all genes, use all the variable genes that we saw yesterday, using dispersion we saw this morning on DSEC is the coefficient of variation, or the genes that are more important for principal component 1 that now you know how to get there, and also principal component 2. Independently of the clustering tendency results, we've also forced patient subdivision using hierarchical clustering that you know, and tested for cluster stability using bootstrapping that you also saw yesterday. In line with previous results, formed clusters were highly unstable using the list of highly variable genes. So approximate p-value of approximate 0, and then you can see that. And that you saw also in the clustering analysis. But there you saw almost 100%. Here you see an example that all clusters are unstable. That means that they are very unlikely to be formed again if you just resample the genes or the samples used for the clustering. So these results indicate that without prior knowledge of patient subdivision, standard gene ranking strategies do not allow clustering of UC patients. And then in another part of the paper, next we perform RNA-seq analysis of colonic samples throughout the differential genes, taking the complete kinetics expression into consideration for p-value estimation using EDGE-R. So based on this, you kind of understand that I used all the samples, of course, for the estimating the p-value. So it's the model that includes everything. And then C method section that I'll go a little bit later. Yeah, and a detailed list of all genes found differentially expressed is available for supplementary dataset 1. And if you look in the method section for the library preparation, now we know from the first day, for example, samples were submitted for library preparation using TrueSeq stranded mRNA library prep kit. So now you know, and then this is poly A selection. So now you know exactly kind of how it was done. And sequencing using HiSeq 2500. And the samples were sequenced using single-end 5-base pair sequencing, aiming for a coverage of 20 million reads. And read quality was inspected with multi-QC using Trimmomatic to trim some other parts, but you haven't done that in this course yet. And further proceeds for abundance estimation using Callisto. That we'll see tomorrow, actually. But in essence, it's exactly the same as you have done with HiSeq. So further data analysis was done in our programming language. Genes with absolute read count less than 5 in at least 3 samples, which is exactly the cutoff that we used, were considered low expression and filtered out. And another thing that Roy mentioned in his analysis is when you do normalization, control for compositional bias. So in some cases, you have some genes that are highly expressed, or maybe there are different cell types that will influence the gene expression in your data set. And this is a normalization that we haven't done in the course because those 2 samples do not have that issue. But we saw that in the other samples of the data set. And to do that normalization, so we used a couple of reference genes, which is this, what also Roy mentioned. So use some genes that are considered the housekeeping to normalize all the other genes. So here, the differences in cell composition that could affect transcriptional pools were balanced by means of removing unwanted variation based on negative control genes using the RUVG function. Roy actually mentioned this one, but we didn't use it in the paper. So yeah, so here I show how I get the housekeeping genes, which is controlling them for correlation between the genes and the library sizes. And then these negative control genes were thus defined as genes with positive Pearson correlation. And the estimation of the unwanted variation were used as covariates here for calculation of differential gene expression. So before this lecture, I mentioned, like, if you have a patient age, you should use that covariate as numeric and not as a group. This is exactly the same case. So RUV-seq, it estimates some values, and those values will be used to correct and normalize each sample individually based on the values. So all of this is put into EDGE-R for differential gene expression. So EDGE-R is specialized in performing time series differential expression by means of GLM, which I already mentioned today as well, where the time points were passed as independent factors in the contrast matrix, thus allowing detection of differentially expressed genes at any given time point. So now you know, again, I used the whole model with all the samples. Genes were considered significantly differentially expressed when the overall false discovery rate, which now you know what it is less than 0.001, and at least one time point had a fold change of above 1.5. And then genes identified in this manner were used for dimensionality reduction by principal component analysis, from which gene contribution to total variance can be calculated. And then later, identification of the gene modules were done by smooth temporal, and this is a little bit more specific. So you have the gene expression over time, so you can actually smooth that rather than have per sample variation. You have like a overall trend over the time course. And briefly, gene-wise log fold changes, or the normalized data, were smoothed using the spline curves and further grouped into modules by using inverse Pearson correlation that you also saw in the clustering lecture as distance, for hierarchical agglomerative clustering with Ward's method, or Ward D2, which is exactly the method that you also saw in the lecture. Functional gene annotation was performed on each gene module individually using gene ontology. Now, I'm specifically saying the 2017 in this case, and the Kyoto Encyclopedia of Genes and Genomes, KEG 2016 libraries in an ENRICH-R package, which is exactly what you did today. So time series analysis using, so this is one of the figures, just mentioning again. So you have metadata group, and then you basically say metadata group and then level everything to day 0. So the day 0 is my control. So everything compared to day 0. And then you write a formula simple as this one. For principal component analysis later, principal component analysis, this is now the results part. So principal component analysis on differential genes, expressed genes. So now you know that I didn't use highly variable genes in here. I used the ones that are already differentially expressed. Revealed that samples displayed a sequential temporal path in PCA space, which is this line, starting on one and another. We observed that over 70% of the variance among the differentially expressed transcript is retained in the first 5 principal components. Now you know that each principal component stores the highest variance in the data set, starting by 1, 2, 3, up to 5. And that variance is literally part of the data, at least part of the differentially expressed genes that we have in the data set. And that means that most of the data is explained by only a couple of principal components, and it's worth to look into them. So here, and then each principal component corresponds to a unique expression kinetics through the time of course of DSS colitis. So if you look in the supplementary file, you see the time course 0 to day 14. And then here is the level of PCA. So how the samples go up and down. And you can see PC1 going like this, PC2 moves in this way. And actually there are all the at least 6 principal components. And then in the latest one, there is not much of a trend, while the other ones, they have something else happening. And later, so for instance, the variance explained by PC1. So now you have an idea what variance explained by PC1 means. Peaked at the acute phase and almost returned to normal levels at day 14, which is basically this plot. Capturing most of the variance related to inflammatory genes that peaked from day 7 to day 10, such as, and then here's a list of genes, which are shown in here. So this is the same thing that you did in the PCA lecture. What is the importance of every gene to principal component 1? And the same thing, on the other hand, the variance explained by PC2 peaked at some point and then returned to normal levels and then corresponds to genes such as A, B, C, and D. And those genes are listed here. So now you know that these genes, what these genes mean. And the latest thing, the last thing actually, is once the paper is published, so then all raw data generating the study was deposited in GSEA. And the code is also available there, is all in the same paper. So you can check on GitHub how the process data of this paper was done, for example. I saw that many papers are starting to do that more often. It was not a requirement back then. And it's becoming more and more common to have all the code and how exactly you get those results done. And that's also one of the reasons why we try to use Conda. So Conda is an environment, for example, nowadays I cannot never recreate exactly the same figures in that paper because we don't have that environment. And that's why we're using that in the course right now as well, to have the same environment or at least similar environments for everyone.